Hi, I'm Danny O'Brien, veterinary neurologist at the University of Missouri College of Veterinary Medicine. And this is a recording of a presentation I gave to the Golden Retriever Club of Kansas City in 2019 about identifying and preventing hereditary disease. So what we're going to talk about in this presentation is first, what is the disease that we're talking about here, neuronal ceroid lipofuscinosis. We're going to talk about how hereditary diseases happen in breeds and how we go about finding the gene responsible for a hereditary disease, and then what you do with that information once we found a mutation. So the poster child for this presentation is a dog named Lexi. Lexi was born as a field trial golden retrieval. She should have been the ideal pup. Her parents came from two different field champion bloodlines. Her father was a grand champion field trial dog. Her mother hadn't won any championships herself, but she came from very good lines and had a very good disposition and obviously a beautiful golden retriever. So initially, Lexi was performing very well in her training. At 6 months of age, she was sent off to a professional trainer, and all the reports were that she was going to be an excellent field trial dog. She was learning fast and performing well. Now, it started to get cold up north where she was being trained. They sent her down to Texas for the winter. But by the time she got back to training again, she wasn't doing so well anymore. She wasn't performing nearly up to the speed that she had been before she went to Texas. Something was clearly wrong. When she got back to her owners at about 18 months of age, it was clear that something was not right. She was restless and anxious. She would pace around. Her coordination was poor. She would bump into things. And she'd have these periodic jerking movements of her head and face, like she was startled or something was scaring her, or she would just make this sudden jerk and rub at her face, like that. Over the next couple months, things got worse. She became demented. She didn't know where she was. She didn't know her owners anymore. All she would do all day was engage in these repetitive behaviors, kind of pacing back and forth or moving back and forth like this, looking at things that didn't appear to be there. She was taken to a veterinary neurologist who examined her and confirmed their suspicion that something was neurologically wrong and performed an MRI to get a look at Lexi's brain. Now on the right here, we've got a picture of the MRI from a normal dog. The gray area that you see in the middle on the top is a cross-section of the brain. The bottom is what's called a mid-sagittal section, kind of a down the middle section of the brain. And what you see here is a perfectly normal brain, where the skull is filled with the brain material. A dog with NCL, however, has a very different MRI. All the white space that you see on these images is fluids that has filled the space where the brain is atrophied away. So there's been marked atrophy of all areas of the brain in Lexi. And unfortunately, Lexi had a very poor prognosis. Her quality of life was poor, and they elected to euthanize her. A post-mortem exam was done, and when they looked at the brain in Lexi, the neurons, which is shown in the middle of this image here, should look more like, there's nothing really normal here, more like that corner of that neuron there, where there should be a kind of a homogeneous looking substances within the cell. Instead, what we see are these speckles of dark purple stuff that have built up within the cell. And this is the hallmark of a storage disease, a disease where material is getting stored in cells that doesn't belong there. In this case, the nerve cells in Lexi's brain. So when we're faced with a disease like this, that is a progressive neurodegenerative disease in a young purebred dog, one question that we always have to ask ourselves is, could this be a genetic disease? And now how do we go about determining whether something is genetic or caused by another disease process? Well, on the MRI, we can look for things like evidence of infections or neoplasia. We'll do a spinal tap to also look for evidence of infections. We look at blood work to see if there's any metabolic cause that might be causing these progressive behavior changes in Lexi. And when we ruled everything else out and we're beginning to get really suspicious that maybe this is genetic, the key is going to be to look at the family and see, is there a history somewhere in the family of other dogs with a similar kind of signs? And if we see a pedigree that looks like this, in this pedigree, it's a little different from the way breeders are used to looking at pedigrees. And we start with the grandparents and great-grandparents on the top and make a pyramid going downward from them rather than starting with one individual and going backward in time like you do for a standard breed pedigree. The squares are the males, the circles are the females, and the shaded in symbols are dogs that had similar signs to Lexi. So when we see a pattern like this, where a percentage, on average about 25%, of the offspring of two perfectly normal dogs are affected, this is a classic pedigree for an autosomal recessive disease. So this would suggest that this is a genetic disease. So once we've answered that question, that yes, this is something that's inherited, what do we do about it? Well, the answer is we want to now find the mutation that's responsible for this disease. Now, we're fortunate at the University of Missouri to have an outstanding genomics team there. Dr. Gary Johnson has been doing genomics before it was a word. He has a number of great people working in his lab. Liz Hansen is a standard Schnauzer and Picard breeder who handles all the communications and data collecting in Gary's lab. And right now, Dr. Natalie Villani is a graduate student working on her PhD in Gary's lab. And the approach we're taking now at Missouri to find genes responsible for diseases is a whole genome sequencing approach. Now, in a whole genome sequence, what we do is we take one affected dog, such as Lexi, and we basically sequence their entire genome. Every single one of the 2.5 billion pairs of DNA molecules in their genome will be read and put into a computer spreadsheet. That's essentially like we're looking for a criminal and we're going to go out and interrogate every single person on the planet to see if we can find the culprit. But how do we go about sorting out of those, 5 billion DNA molecules, which two are the actual culprit causing this disease? Well, to do that, you need two things. First of all, you need a lot of computer power because we've got 5 billion bits of data here that we have to analyze in multiple ways. And we also need a Schnabel. Dr. Bob Schnabel. Bob is our bioinformatics guy who's helped us get the computer program set up to do this kind of analysis. So what we do with that information is we take the whole genome sequence from a dog like Lexi and we compare it to whole genome sequences from over 100 other dogs that we have sequenced for other reasons. Now, some of these dogs have diseases, but they have different diseases. So there should be a mutation, a change in the sequence in a dog like Lexi that is not found in any of these 100 other dogs. Because while they may have a different disease, they would have a different mutation in a different gene. So it won't be the same as what's in Lexi. And so what the computer programs that Bob has generated do is they compare every single DNA molecule in Lexi's genome with this corresponding molecule in these 100 other dogs and pull out what's different about Lexi, because one of those things that are different about Lexi is going to be the culprit in her disease. So this is the result of one of these sequences. The top line here is what we call the reference sequence. That's the normal sequence from the original dog that was sequenced as part of the Canine Genome Project. The bottom lines here, won't get into the details about why there's multiple ones down here, are the sequence from our affected dog. And you can see that the majority of things are the same between the affected dog and the reference fingerprint sequence until we get to these two DNA molecules right here, which aren't there in our affected dog. So what we have is a deletion. Something has knocked out these two molecules in these dogs. And this is in a gene called CLN5, which stands for ceroid lipofuscinosis neuronal number 5. And this is a gene that is known to cause lysosomal storage diseases. What we do next is then we go back to the family and see, is the segregation of this mutation that we found in the affected dog consistent throughout the family? Because if it's the culprit, all of the affected dogs should be homozygous for that deletion. The parents should be heterozygous for the deletion. And all the normal offspring should either be heterozygous and carriers like the parents, or they could have gotten the luck of the draw and get 2 copies of the normal gene. So we're going to label these individuals with their genotype, the combination of genes that they have. A DEL will suggest that they've got a copy of the gene with the deletion, whereas a WT is a copy of the normal gene, which is often called the wild type. And what we can see is that everything is perfectly consistent. The grandmother was a carrier of the deletion. She passed that on to her 2 offspring, which were crossbred. And you can see in the pedigree how every single affected dog is homozygous for the deletion. These dogs were not available for sampling, while every normal dog is either homozygous for the wild type or is also a carrier. I should mention that this is not Lexi's pedigree. This is a pedigree of another affected dog who was actually the one that was sequenced for this study. Then to get further proof that this is indeed the culprit, we look at a broad sampling of DNA samples from the breed to make sure that there are not exceptions out there. There are not dogs that are homozygous for this deletion in the gene that are normal. And out of 1,000 random DNA samples from golden retrievers, we identified 8 carriers, and we also found 1 other dog that was homozygous for the deletion. And when we contacted the owners, they told us that she had died of a very similar disease. We also test a random sampling of DNA from other breeds, 99 other breeds, and we didn't find the mutation anywhere in any other breed. So this is very strong evidence that this is the cause of the disease. So what is CLN5? As I mentioned, this stands for ceroid lipofuscinosis neuronal number 5, the 5th form of this disease that has been reported. In human medicine, this is referred to as Batten disease. And we're fortunate also at the University of Missouri to have over at the med school Dr. Marty Katz, who is one of the world's expert on this particular form of disease in children. And what differentiates this disease from other types of storage disease is that if you take a slide of the brain tissue from an affected animal and you put it under an ultraviolet light under the microscope, this material, the storage material that is built up in the cell, fluoresces. And that is the hallmark of neuronal ceroid lipofuscinosis. So what are we talking about when we talk about a lysosomal storage disease? To understand lysosomal storage disease, you have to understand how the cell manufactures and recycles proteins. The basic building block of proteins are amino acids. And you can think of them like different colored Lego blocks where each amino acid has its own color. And these different amino acids are put together to form a 3-dimensional structure that's going to be the protein that the dog needs to perform some function. And even though we start out with a fairly simple, set of building blocks, you can put these together to make a very complex structure, a complex protein that does what the cell needs to do to function in the body. Now, because the ability of the cell to manufacture the proteins is perfectly fine in dogs with lysosomal storage diseases, The dogs develop normally. Everything is built the way it should be. So the proteins are there, they're functioning the way they should, and the dog looks perfectly fine for a variable period of time. 6 months, a year, sometimes a little longer, sometimes a little shorter. But nothing lasts forever, and the proteins in the body are going to get damaged. Something happens to them. They get broken, they get misfolded, something goes wrong with them, and now they need to be disposed of. Within the cell, there's an organ called the lysosome, and the lysosome is the recycling center of the cell. And so what the lysosome does when it detects a protein that's damaged and need to be disposed of in the cell, it engulfs it. It swallows it up and takes it inside. Now, inside the lysosome, there are going to be enzymes that can break down this protein. They can pull off these amino acids. And for each bond between different amino acids, there's a different enzyme. So you need a variety of different enzymes to stepwise break this protein down and get it down to its component parts. And this process just keeps going on until all the amino acids are broken down, and then they can get released back into the cell where they can get reassembled into a new protein. What happens in lysosomal storage disease, however, is one of these enzymes doesn't work like it should. So in the case of the CLN5, we had that deletion of the AG in this little component of the enzyme here, and that leads to a defect in the enzyme that can affect the ability of that enzyme to function. And if that enzyme can't function, then the ability to break the bond of that red amino acid with the other amino acids doesn't work, because no matter how hard the enzyme tries, it can't break that bond. And even though only that enzyme is affected, in order for other enzymes to work, you have to get past that red block step in the process. And the other enzymes can't do that. They can't do an end run around it. And now we have a component of that protein that cannot be broken down any further. And until it's broken down, the lysosome can't get rid of it. It can't recycle it. And so what happens is that fragment builds up within the lysosome. It gets stored in there. And eventually it builds up to the point where it interferes with the function of the cell. The cells start to die, and we get neurologic disease. And these purple granules that we're seeing in the neurons in Lexi's brain are that storage material within the lysosome that is built up and will ultimately kill this nerve cell off. So hereditary diseases are common in dogs, but not for the reason you might think. When a dog wins championships, when they win field trials, it's because they've got the right stuff. They've got what other breeders want. They are what a golden retriever wants to be. So it's only natural that when a stud wins championships, that it's going to get widely used, because all breeders out there want that same stuff in their lines. The problem is, every dog out there, every person has mutations in their genome. Mutations happen. And it's estimated that every person has at least a half a dozen potentially disease-causing recessive genes in their own genome. And it's no different with dogs. So this grand champion that has all the right stuff to be a great field trial dog also has some disease-causing recessive genes in his genome, along with those desirable ones. So it's only natural that this stud is going to get widely used. He's going to go out there and breed a lot of offspring. Now, in the first generation, everybody's happy because they've got this grand champion blood in their lines now, and they're looking forward to producing good dogs. The problem comes in the F2 generation, when these dogs' offspring start breeding to each other. That's when we're going to see what recessive traits this guy was carrying. Now, when the first dog is born, this first black box up here, people probably don't think too much of it. You know, a dog has a problem. Okay, problems happen. You know, one dog, you know, not that big a deal. But if this dog gets widely used as a stud, there's going to be a lot of carriers out there in the population. And as you look at generation after generation after generation, breeders start talking to each other. They start saying, oh, yeah, I had a couple pups like that too. I hear that John did too. And then you start to realize that there is a problem in the breed. And now, though, it's too late to put those genes back in the bottle. They're out there in the population. And remember, these are the good breeders, the ones that are trying their best to produce the best dogs. And that's why they went out there and got this field champion blood in their bloodlines. So bad dogs happen to good breeders. Nobody is trying to produce dogs like this. Now, when there's a problem in the breed, there are two approaches you can take to it. One is the ostrich approach, where you hide your head in the sand, pretend the problem isn't out there, and hope that it will go away. The problem is, if it's widely disseminated in the breed like this, it's not going to go away. It's going to be a continuing problem. The better approach is to fight the problem head on, to tackle it and get the tools to solve the problem rather than just ignoring it. And the tool that you're going to have in a situation like this is going to be a DNA test. Once we've found the mutation, we can develop a test to detect that mutation. Now, here's an example of what this test might look like in one way of running it. So what we have over here is a dog who has 2 copies of the mutation. Ignore that light line there, because there will be some light lines that show up on these things. But the dark line there is the mutant allele, the mutant gene that causes disease. So when we see a dog that has 2 copies of the mutant one, no copy of the normal one, we know that this is a dog that's homozygous for the mutation, and he's at risk for developing this disease. If we're lucky and we get a dog that has 2 of the normal genes, then we say that dog's clear. It doesn't have a mutation. There's nothing about this DNA test that causes any concern. As a breeder, what you're looking for is to identify who is the carrier. Who is the dog who has 1 copy of the gene that's causing the disease and 1 normal copy? Because in a recessive trait, if you breed 2 carriers together, that's when you're going to get disease. Now, the thing to recognize about a DNA test is that it's very sensitive and very specific for the mutation, but not the disease. So, for example, if we look at this particular gene, CLN5, we also see ceroid lipofuscinosis number 5 in Aussies and Border Collies. So they have the exact same disease, they have the same storage material, the same enzyme isn't functioning. But when we look at the mutation in the golden retrievers, it's this deletion. The mutation in the Aussies is a little different than the Border Collies. It's just a change in the amino acid. There's not a deletion of that DNA molecule. There's just a wrong one there. But the result is the same. The enzyme doesn't work and the dog develops disease. The trouble is, if you do the test for this mutation in a golden retriever, it'll look like the golden retriever is fine because it doesn't have that mutation. It's got this one. So you need to be doing a DNA test that's for that particular breed. And then what you're going to want to do as a breeder is to test your breeding stock. And with the DNA test, you can identify who's a carrier, who's clear of the disease, and then you can just eliminate the carriers from the breeding population, right? Wrong. That is not the thing to do because there are problems with just eliminating the carriers. Okay, so, all right, so let's say 1/3 of the population has carriers. Well, we can eliminate them. Golden retrievers are a big breed. You've got plenty of, gene pool to swim in, even if you eliminate 1/3 of the breeding stock. The problem is going to be what's going on in those other lines. Are a lot of the dogs in those other lines ichthyosis carriers, for example? Okay, well then we're going to have to eliminate those guys from the breeding stock too, otherwise we're going to have ichthyosis. Okay, so now we've eliminated, another chunk of our breeding population. And maybe these guys have a high incidence of PRA carriers. Well, we eliminate those. What we're doing here is we're making a bottleneck in the gene pool. We're giving you the breeders fewer and fewer choices in terms of who they can breed to, because everybody's carrying something. There's going to be a bum mutation out there in every line. And there's been a study done by Claire Marsden that we helped with that looked at what really contributes to deleterious genetic variation in dogs. It's not inbreeding. It's the bottleneck of a breed, a winnowing down of the breed pool to too small of a selection that causes the most deleterious effect on the genetics of that breed. So as we're thinking about getting rid of a disease like this, we need to think not just about the individual, but about the population and what's good for the genetic health of the population as a whole. And to do that, we want to keep those carriers in the gene pool. Also, we don't want to throw out the baby with the bathwater, because there's a reason this guy won all those field champions, because he does have a lot of great genetic material that you do want. And if you eliminate him from the lines, you eliminate all that good stuff as well. So what we're going to do is we're going to want to keep those carriers in the breeding pool and just use them wisely. If we never breed two carriers together, then we will never produce an affected pup, because you have to have two copies of the defective gene in order to have disease in an autosomal recessive trait. So if we know who is a carrier and who isn't, we can use a carrier, we just always have to breed them to a dog who is clear. Now, as we're looking at these pups and trying to decide who's going to be our future breeding stock, we consider the genotype as one consideration as we're trying to make that decision. So if these two pups here are pretty much equal in terms of what their desirability as a future breeder is, this one's a carrier and this one isn't, then it's a no-brainer. We keep the clear pup, and that's then going to be our future breeding stock. We can breed her to anybody and not have to worry about it. But if this guy over here is head and shoulders above these others in every other respect, but he's a carrier, then so what? We still use him. We just make sure that he never breeds to another carrier and will never have an affected pup. Over time, we try and select more and more clear dogs as our breeding stock, decrease the incidence of that allele out in the population, and get rid of the disease. So the test for CLN5 mutation in the golden retrievers is available through OFA. Dr. Johnson's lab at MU does the DNA testing for OFA. But we have no conflict of interest here. We make nothing from these DNA tests. Any proceeds from the DNA testing that Gary does goes to the research. Nobody makes any money off of it. It's purely a not-for-profit enterprise, both for OFA and for the MU where we do the testing, whereas other labs are commercial enterprises that are making money for shareholders. There are some other golden retriever diseases that we're currently studying. We are seeing what we call a remitting ataxia, where there are some pups born that have fairly poor coordination when they're young. You can see how this guy's kind of goose-steppy and kind of wobbling like a drunk sailor there. When they try to walk downstairs, you can see that they seem to be having hesitancy about stairs. Sometimes people will think they have a vision problem because they're so hesitant, but it's a coordination problem. They're scared of the stairs because their coordination isn't very good and they might fall. They can also have these little head tremors where as they try to focus on stuff, they get this kind of little wobble head kind of look. The good news about this disease is it does seem to get better with time. They seem to grow out of it, if you will. But still, these pups are uncoordinated when you're trying to place them in homes. And so we're looking to find a solution to this one. So if you have pups that seem to have this kind of coordination problem, we'd like to hear from you. We've also seen a case of a dog that had what appeared to be a hereditary peripheral neuropathy that caused weakness of his lower part of his limbs. You can see how he's kind of collapsed down on his carpus there. And eventually it gets to the point where he can't use those front legs at all. It's very unusual to have a front leg specific paralysis like this. So we're trying to figure that one out as well. There's also another form of lysosomal storage disease out there. I presume there's a mutation in one of the other enzymes that's necessary to break cells down, and we're working on that one. And the last disease we're working on is one called Ehlers-Danlos syndrome, which can make for funny pictures when you look at these dogs, but it's not very funny, really. This is a disease of the skin where the elasticity of the skin is abnormal. And like I said, you can make funny pictures of the dog by stretching his skin out like that, but often the skin is easily torn. These dogs are subject to injuring their skin. Their joints are equally lax, and so they have joint injuries, joint problems. And we know that there are at least 2 forms of this disease out there in golden retrievers, and we're trying to get that one solved here as well. So this work was supported by part by the Canine Health Foundation, and we appreciate their support as well as the Golden Retriever Club's help in solving this problem. What I wanted you guys to get out of this was to recognize the signs of storage disease. In golden retrievers in particular, this one form is called neuronal ceroid lipofuscinosis. You should appreciate how we show a disease is hereditary and find the mutation responsible for that disease, and understand how this mutation leads to the signs, how this enzyme deficiency leads to this storage of material within the lysosome that ultimately damages the cell and causes this progressive degeneration after a period of normal development. Now that we've got a DNA test for this, you need to use the test wisely and develop wise breeding strategies to avoid the disease but maintain desirable genetic diversity within the breed, to avoid those bottlenecks that are just going to trade the devil you know for the devil you don't.